We have Josh Bouma here, and he is from the Puget Sound Restoration Fund, who is overseeing this restoration project with the Pinto Abalone. He is a shellfish biologist, scientific diver, and abalone recovery program director at PSRF. He leads Pinto Abalone conservation aquaculture efforts, research, and field programs aimed at recovery of this charismatic and unique, but severely threatened native mollusk. Josh also manages the Port Madison Community Shellfish Farm for PSRF. He earned his master's in aquatic and fishery sciences from the University of Washington, and he enjoys diving, eating seafood, growing things to eat and drink on his small hobby farm, and spending time with his family. So let's welcome Josh.
Good afternoon, everyone. I really appreciate being here. Thanks for the invitation, and thanks for that introduction, B. Yeah, I'm really excited to be here to give this talk to you guys today. This has been an ongoing program for a number of years that the Port Townsend Marine Science Center has been a great partner in. It started, over a decade ago with citizen science and education with high schoolers in the area, and it's continued in recent years with the Port Townsend Marine Science Center becoming a key partner in our satellite nursery program and culturing some abalone for release to the wild, and continued engagement and outreach. So really appreciate the partnership, and I'm happy to be here. I'm going to give a talk today, a fairly broad talk, kind of summarizing where we're at with recovering this species in Washington. I'm going to talk about the use of conservation aquaculture to give the Salish Sea's largest rocky reef grazing marine snail a population boost. I'm happy to keep things a little informal if you guys would like. If you have questions about a figure that you don't understand or a term that I need to clarify, just don't hesitate to interrupt so that we can all stay on track. And maybe if you have bigger discussion questions, we can save that for the end. Okay, well, let's dive in. Acknowledgements are often saved for the end of a talk, but I really want to emphasize the strength of the collaboration in this program. We have a really diverse group of state and federal agencies, nonprofit organizations, universities that have been key to really propelling this program ahead. And so I want to just acknowledge that collaboration is really the strength of this program. So that's always a good way to start. To give you guys a sense of where we're headed with the presentation today, I want to start by talking about what is an abalone and why are they important. And then we'll spend maybe 10 minutes or so talking about the status and decline of abalone here in Washington State specifically. And the bulk of the talk, though, is going to focus on conservation aquaculture and our abalone recovery program. So first of all, what is an abalone? I'm sure many of you are already familiar, but for those of you who aren't, abalone are a large charismatic marine snail. They're in the phylum Mollusca and class Gastropoda. They're all found in the genus Haliotis, which stands for sea ear. I'm sure you're all very familiar with that really recognizable mother of pearl that you see on the inside of an abalone shell. There's actually 50 species worldwide, almost all of which are in decline because of harvest pressures. Oops, wrong button. I was just going to highlight this picture here. This is Monterey in the 1930s, and that's a pile of mostly red abalone shell during actually kind of early on in their commercial fishery in California. There's actually 7 species of abalone on the West Coast. 2 of which are actually ESA listed species, the white abalone and the black abalone. The other 5 species are all either threatened or species of concern. Pinto abalone specifically is the only abalone that we have here in Washington State, but it also has the widest range of the West Coast species. So it ranges from Baja, California, Mexico, all the way up to Sitka, Alaska. There is a subspecies, the threaded abalone, that some people think is maybe separated by point conception, but genetically speaking, threaded abalone and pinto abalone are the same species. The habitat for abalone in general, Pinto abalone specifically, are these really dynamic, current influenced rocky reef habitats, also associated with kelp beds, primarily Nereocystis, bull kelp. The depth for Pinto abalone is usually 60 feet or less, although they have been reported up to about 100 feet. Most of our work happens in the shallows, 0 to 40 feet. These are herbivorous grazers. They feed on benthic diatoms and bacteria that biofilm on the reef, and they also take advantage of drift macroalgae or kelp. And these are dioecious broadcast spawners. So there are specific males and females that release their gametes into the water column for fertilization. So the life cycle of these guys is fairly unique. As I mentioned, the adults, these guys here, males and females, are going to release their gametes into the water column, hoping for remote fertilization, which is why this is a really key characteristic of abalone. They really need that dense, those dense aggregations for successful fertilization in the wild, and that's going to come up quite a bit in this talk. Once those eggs are fertilized, this developing egg is actually negatively buoyant and will sit on the rocky reef for about 24 hours as it develops, and it hatches into free swimming or planktonic larvae. It has a trochophore and a velager life stage. A unique characteristic of these guys, they're actually lacithotrophic, which means non-feeding. And so they're really reliant on the yolk reserves that they get from their mother or from the egg. And this also drives a fairly short larval period, 7 to 10 days in the plankton. And this is unique. And it also potentially means that abalone have a much less of a distribution than other planktonic larvae who are in the water column for much longer. After 7 to 10 days in the water column, they're going to settle out as post larvae when they see that nice, clean, coralline encrusted rocky reef habitat. They're very chemically cued into that. And then when they hit the 5 millimeter mark, they kind of transition their feeding behaviors from that biofilm more so onto macroalgae, and that's when they become a juvenile. And when they hit about 2 to 2.5 inches, that's when they're reproductively mature and become an adult. I'll grab a quick sip of water. Should have brought an easier water bottle to drink from. I'm going to have to take lots of drinks. Okay, so that's some of the biology. Why do we care specifically about Pinto abalone? Well, this is a unique native species of the Pacific Northwest. They've been culturally important to indigenous populations that have used them as a food resource, but also that shell has been a form of currency and was used in regalia for indigenous peoples as well. They also have been economically valuable. There was dive tourism based around this species, and there was a recreational fishery up until 1994. It's estimated that somewhere between 35 and 40,000 abalone were taken from the San Juan Islands at the peak of the recreational fishery. And I'll just make note that populations here in Washington never supported a commercial fishery for pintos. It was always just a sport dive fishery. Most importantly, I want to talk about abalone as ecosystem engineers. Their grazing behavior is going to maintain the rocky reef substrate, and that creates habitat for the settlement of other invertebrates and kelp. So they really are tied into this larger idea of the food web on our reefs here in Washington waters. Grazing behavior of abalone is going to support settlement and growth of a good dense kelp canopy, which then supports forage fish, which of course is a food source for salmon and orcas. So it's not unreasonable to think about abalone as a really important critter in terms of the overall food webs here. Well, let's spend just a couple of minutes talking about the population status here in Washington State. I just want to emphasize that the range here in the state is actually from the San Juan Islands in the north all the way out across, through the Strait of Juan de Fuca as far as Nia Bay. They're not really found on the open coast here in Washington. But most of the work that we've done, including population surveys, broodstock collections, our restoration program, has really been focused on the San Juan Islands and surrounding areas. We haven't moved into the straits as much yet, but that would be part of the plan moving forward. I'm going to talk about this figure here for a couple of minutes. As part of the coming to a better understanding of population abundance in Washington, the Department of Fish and Wildlife set up 10 permanent index stations here in Washington. That work began in 1992, and those 10 index sites have been surveyed regularly over the course of the last 25 plus years. And what you see here is a 97% decline in abalone abundance between 1992 and the most recent survey, which was 2017. This green line up here is the kind of the minimum fertilization threshold for broadcast spawners. And you can see even early on in this study, 92, 94 were some of the first years. We were already hovering right around that minimum density necessary for successful reproduction here in Washington. The first year of the survey in 1992, there were close to 400 abalone counted across all sites. 2017, 12 abalone were counted on those 10 sites. 5 of the 10 sites have been extirpated or no longer have abalone on them. It's been a while since we've done these surveys. I'm going to participate with Fish and Wildlife in February. We're going to redo all 10 surveys and kind of see where we stand at this point. I also just wanted to add this line here marking the recreational fishery closure in 1994. I think what WDFW hoped and assumed is that if they close this harvest pressure on abalone in Washington, you might see a stabilization of the population here and then a slow recovery. But the opposite of that occurred, and we continued to see this precipitous decline to where we're at now. On this figure here, I'm going to talk a little bit about how recruitment is failing. So when I talk about recruitment, I'm referring to the ability of a population to successfully reproduce and then have the juvenile offspring kind of survive and then replace that aging population. So recruitment is clearly failing here in Washington. This is your typical size frequency distribution. You can see the shell length of the animals counted on the x-axis and the frequency of observation here on the y-axis. In the blue, these are the early surveys, 79 to 81. Average shell length was about, let's see, 98 millimeters. By the time we get into this survey period between 92 and 96, average shell length was 105. And up here in 2000 2013, we're all the way up to about a 120, 125. So you can see clearly we have an aging population that's not being replaced by juveniles or new recruits. I think the smallest animal we observed in the 2017 surveys was 83 millimeters. So we're just not seeing those small new recruits in the population anymore. All right, so what caused this decline in abalone here in Washington? There's a number of proposed contributing factors. The first is that the permitted recreational harvest was too high. Secondly, it's very possible that illegal harvest was more abundant than estimated. In the late 80s, early 90s, you had a real boom in commercial fisheries for sea cucumbers and sea urchins. And it's thought that many of these harvesters may have been taking abalone illegally on the side. In fact, there was in 1994, a commercial harvester was arrested for poaching Pinto Abalone. I don't have the picture in this talk, but his boat was actually named the Abalone Maid, M-A-D-E, Abalone Maid. He was not shy about bragging how he was making his money taking abalone illegally. In addition to that, we now have recruitment failure, which I just described. We call that the Ali effect. So when you have a population of broadcast spawners that has been spread out too thin, they're no longer in a density where they can have these successful reproductive events. You get recruitment failure. That's what we like to call the Ali effect, and we're certainly seeing that here in Washington. And now, environmental factors may be affecting reproduction, larval, and juvenile survival. Things like salinity and temperature changes, increased sedimentation, and ocean acidification is a big one as well that we're doing a lot of research on. So NOAA actually received a couple of petitions in 2013 to federally list pintos as an ESA listed species. They went through their year-long review process. They did a very thorough job. In 2014, they determined that Pinto abalone are not warranted for ESA listing. And that's fair because we have a wide-ranging species from Alaska to Baja, and not a lot is known about populations in other parts of their range, whereas here in Washington, it's our only species, so it's been thoroughly monitored and researched over the years. In order to get an ESA listing as an invertebrate, you have to show loss across the range of the species, and that's not necessarily the case for pintos, just because there's that lack of knowledge of what's happening with pintos in other places in their range. Given that, the state of Washington has listed pintos as a state endangered species. That listing happened in 2019. And following that listing, we developed the recovery plan for Pinto abalone in Washington that was published in March of 2022. This is a really interesting document if you guys are, have some time and want to get deeper into what a recovery plan looks like. It talks a lot about the history of the program that we've been running for over a decade and a half now, but it also gives the delisting criteria, which I think are really interesting to give a look at. All right, so given what I've explained so far with the biology and status of the species, I kind of want to get into the meat of the talk here and talk about conservation aquaculture as the methodology that we think is going to have the best chance of recovering the species here in Washington. Given what we know, it seems like human intervention is probably the best path forward. And so we've spent a lot of time over the last 15 years developing this program based on conservation aquaculture with the primary tenet to do no harm. We want to have as little impact on what's left of the wild population as possible. So we've developed this captive rearing program and then used that program for juvenile outplanting at restoration sites throughout the San Juan Islands. The Hatchery Culture program actually started at the NOAA Mukilteo Research Station in about 2007. So it was initiated by Dr. Carolyn Friedman at UW and folks at NOAA and WDFW. But we have transitioned the program completely over to the NOAA Manchester Research Station. And in 2015, PSRF opened the Kenneth Chu Center for Shellfish Research and Restoration. We have a hatchery building, a greenhouse, and nursery building there. And in that nursery, we have about 100 culture tanks that we use for growing out abalone for restoration. This is at the NOAA Manchester Research Station, just south of Bremerton. Yeah, it's a really great collaboration between PSRF and NOAA. And we not only do work on abalone there, but we culture Olympia oysters for restoration and do research on basket cockles, sea cucumbers, geoduck, native clams. And we have a kelp lab there as well, where we're culturing bull kelp and sugar kelp for research and recovery. It's a really cool place, and we're happy to give tours if anybody wants to come down and take a look at the facility. I highly encourage that. All right, so one of the key components of any captive rearing program is our broodstock management. We want to maximize genetic diversity. Therefore, we try to collect new broodstock annually. And this is an effort between PSRF and WDFW divers. We usually spend a couple weeks each late winter looking for new broodstock for our hatchery program, and we're only collecting reproductively isolated singletons. So anytime we find an aggregation, meaning 2 abalone or more that are within 5 meters of each other, we're going to leave that aggregation intact. So we want our broodstock collections to have as little impact on wild populations as possible. It can be difficult and time consuming to find singletons. It's a lot of dive time, but it's worth the effort. And in a given year, we may bring anywhere from 15 to 30 new animals into our hatchery program to support genetic diversity. So once these animals are in the hatchery, we put them through a fairly specific broodstock conditioning program with temperature control and a feeding regimen. and they're induced to spawn. This is a female here. You can actually sex these animals underwater, which makes it really, you know, we try to have little impact during collections too. And if we know we need 15 females and 3 males, we can actually sex animals before they're even removed from the water and brought up to the boat. So we can identify females by this kind of bluish green gonad and males by this beige cream colored gonad. And this is a female who's just been induced to spawn. You can see the stream of eggs coming from that animal there. One of the another key component to the program is producing first generation hatchery crosses. We're only going to ever outplant F1 generations from the hatchery. We would never take an F1, spawn that animal, and then use F2s or F3s or beyond as part of our recovery program. We don't want to have any kind of hatchery influence or hatchery selection in this process. So we're always going to use wild collected adults. We're going to create these single parent crosses, which you can see in this. This is our whiteboard at the hatchery on a spawning day. You can see on one side you have males, on the other side you have females. And we're trying to, with each spawning event, we're trying to fill out this partial factorial matrix here. Ideally, every one of these boxes would be checked. That'd be a really nice genetically diverse, you know, hatchery day. But it never quite works that way because, you know, we never get as many eggs as we would hope for, and not every animal is going to spawn for us. So you can kind of see what we've done here. We've had a number of males and females that have spawned, and these are the crosses that we were able to produce that particular day. So throughout a spawning season, we're trying to fill out as many of these spawning matrix. We're trying to do as many of these crosses as we can. We've developed these systems for rearing larvae. You can see a velager in the hatchery here. That's about a 250, 275 micron larva. And then this is a settled post set. This guy's about a 0.5 millimeter, maybe 3 weeks old. And you can see this biofilm that the animal's grazing on. We culture 3 specific benthic diatoms in the hatchery that we've kind of determined to be the optimized early feed for these guys. And then our captive rearing includes juvenile grow out in those 100 culture tanks that I described earlier. And you can just see a number of different sizes of animals here. One of the things we've really pursued the last several years is expanding our capabilities in hatchery production. And that means bringing on new partners and collaborators. And we have 2 partners who are now acting as satellite nursery facilities, which is really cool because it gives us expanded capacity, but it also gives us a bit of a buffer and protection against things potentially going wrong at one place over another. So this has been a really important step for us. And the Port Townsend Marine Science Center has been a key partner. Allie is here today and is leading this effort for us at the Science Center here. And I'd say there's been several thousand animals reared here in Port Townsend that have now been released in the wild, and another batch currently being cultured that is due to go out in March or April. So this has been a really productive step forward in the program, being able to add culture at other facilities. Our other partner in satellite nursery efforts is the Seattle Aquarium, and this has been a big step forward as well. The aquarium actually reproduced the same system that we have at Manchester, but cut it in half. So they have a 40 tank culture system. Right now it's sitting on the pier, which is not ideal. It was intended to go at their new offsite animal care center. This whole system is going to be deconstructed at the end of February and rebuilt at the ACC. Which will be a climate controlled indoor environment and should really give us a big boost in production. It's been a little bit tricky doing culture out on the pier in Seattle. We've had some issues with ambient air temperatures and cold snaps and heat waves and all kinds of things really messing with the culture system there. So we're hoping to take some big strides forward at the aquarium this year. They're also going to contribute their dive team to outplant efforts, and eventually the aquarium will be taking on their own recovery sites. So they should be able to find and set up and outplant and then monitor their own network of recovery sites. I added this figure just to give you a little idea of what's happened in hatchery production over the last 4 years or so. There's a lot of numbers here, but I guess mainly what I'd say is you can look here and kind of see how many larvae have been set in our system by year. And you can see there's still a lot of variability. There's still a lot of things we're working out. And I guess this is typical in shellfish aquaculture. It can be unpredictable. The other thing to look at is the genetically distinct crosses that we produce each year. So you can see, 28, 28, 27, 20. These are unique crosses that are produced year by year, just contributing to that overall genetic diversity of animals that are leaving the hatchery program and going out into the wild. Another thing I'll mention too, when you look at the number of larvae set, and then I don't have it on this figure, but the actual number of abalone outplanted to the wild averages between 5,000 and 10,000 per year. You can see our survival from healthy larvae to sending them out into the wild is less than 1%. So there's, as a broadcast spawner, that's just part of the kind of the biology and the life history of these guys. There's very little survival from egg to adult. All right, so I'm going to transition now and spend the last part of this talk. Let me just check my time here and see how we're doing. Yeah, I think we're doing okay. I want to spend some time just describing to you guys what happens after these animals leave the hatchery and get added to our recovery sites in the wild. So we'll spend some time just talking about how this strategy is working out for us. To date, we've released 45,000 juvenile abalone to sites in Washington waters. And our primary objective in releasing these animals is to use healthy, genetically diverse abalone to create aggregations in the wild that grow and survive to reproductive maturity, creating these self-sustaining populations across their geographic range. To date, we have 27 recovery sites. We started in the San Juan Islands specifically, but we actually have sites in San Juan, Skagit, and Island County now. Before the abalone leave the hatchery, they're certified healthy and disease-free. We work with Ralph Elston as our pathologist. So he ensures that we're sticking to that tenet of doing no harm in the wild by releasing healthy animals. A subset of the animals are typically tagged. I'll talk a little bit more about that. And then, of course, we stick to those conservation genetics priorities. And to date, we've released around 200 distinct genetic families. So we feel like we've done our best to really maximize those crosses that we've produced. And then the sites are monitored on an annual basis for survival, growth, and movement. And those are dive surveys that are done by PSRF and WDFW dive teams. This is just kind of a little schematic of what an outplant site looks like. We have these permanent markers on the corners. When it comes time to survey, we can extend a survey tape around it, and then we'll actually divide the plot into 5 lanes with lead lines. And then a diver will exhaustively work his way back and work his or her way across this lane and count and measure all the abalone that are seen. After the first 9 years or so of doing this work, so our first outplants were in 2009, and we've released animals almost every year since then. After the first 9 years or so, we decided it was time to take a real good look at where our successes and failures were. So we worked with a number of different people who had been involved in the outplant work, including some modelers. And this paper was published in 2018, documenting the survival of hatchery origin Pinto Abalone released in Washington waters. This is actually a good image here of what some of the tagged abalone looked like. For the first 9 or so years of the program, we used these unique identifiers. So both color and number identified unique individuals by site. And that became really important to do the analysis for this paper, looking at marker capture data, movement and survival and growth of animals over time. We're not using that type of tag anymore. I think we've learned what we can, and it's I think we've run out of unique numbers and colors at this point. So what did we learn by taking a deeper look at survival? This, again, I'm just going to talk about maybe the first dozen or so sites that we developed. And you can see this might be better labeled as first year observation. So this is what we saw on diver surveys 1 year post outplant. At these first 4 sites, we observed anywhere from 23 to about 5% of the animals that were released 1 year later or 1 year after. And then you can see in 2011, low survival, Jubilee. Actually, I think we recovered 1 abalone on the site a year later. Better results in 2015, 2016, 2017, again, lower observation rates than we would have hoped. So this, I mean, this really says something about what's happening from year to year and maybe what's happening from site to site. So what happened to all these missing abalone? Well, some of them died, but we only recovered 1 to 5% of the empty abalone shells of what was put there originally. So it's very possible that some of these shells are being swept away by currents. They end up in the stomachs of fish, things like that. So we're obviously not going to recover all the shells. Alternatively, these animals could have just left. And this is for restoration purposes, is there really a difference between an animal dying and an animal leaving? If an animal wanders away, it's kind of removing itself from that potential future breeding population. So do we consider those wanderers to be basically dead to the population? This is a really interesting one. I think the answer is no. And the way we figured this out, we started doing perimeter surveys. In addition to surveying the 5 lanes of the outplant plot, we added an additional survey lane all the way around the plot. So that almost doubled the survey size at any given outplant site. And sure enough, we're finding animals wandering off into different places, and not just singletons, sometimes groups of animals are wandering off. So if we're not seeing them in the plot, we can't necessarily say that they've left and are no longer part of that future, breeding population. Lastly, maybe we just can't see them. And we're kind of getting at this one by doing repeat surveys on some sites. And especially with sites that have tagged animals, we can do a survey one day and we may count 100 tagged abalone. We can go back a week later and do the same survey and we may recover 125 abalone, but some of those may be different tags than what we saw the first time. So over the cumulative time of maybe doing 5 or 6 repetitive surveys in a month, you can actually get a more accurate count of what's actually surviving on the plot. We've done that a number of times, and we estimate that our 1-year observation rate may only be 20 to 30% of what's actually on the plot. We've also confirmed that by doing, on a few very small pilot scale sites, we have done non-invasive, our typical non-invasive survey method, and we've followed that up by doing a more invasive survey method where we're picking up small boulders, flipping them over, looking in cracks and crevices. And when we do a more invasive evaluation, we are similarly coming to that same estimate that we're probably only seeing 20 to 30% of what's surviving in a standard dive year survey at year 1. So another way to look, another metric that we've developed to determine success on a site is to look at how many have reached reproductive size. So we're going to call that outplant success, and we're going to look at the current number of animals that have reached that 50 millimeter reproductive size divided by the total number of abalone that were outplanted to the site. Again, this is that same dozen or so of the sites that are some of the earliest in our recovery network. And you can see, oh, and I should point out too, you have both the on-plot and those perimeter animals that I described in those additional surveys. Those are combined on these bars. At a site like Omaha, in a given year, you may see 10% of the total number of animals that were outplanted have actually reached reproductive size, and we can safely assume that those animals have now created a breeding population. So we'd call that a really good site. Then again, you have other sites where you have very few animals that have survived and reached that reproductive size, and we would call those failed sites. I will say that a lot of the newer sites aren't included in this graphic because they just haven't had as many overseedings, and those animals haven't really had enough time to reach reproductive size like our earliest sites have. So hopefully that all makes sense. Yeah, question. I can't tell. Yeah, I'm going to get to why. We actually don't know why, but I'll talk about that. These sites are all in the San Juan Islands, and I can say that we try to be really careful about, like, when we're presenting material at workshops and conferences, we never have maps that show where sites are, just to try to be as careful as possible. But Omaha is a really cool site in the San Juan Islands. And since I'm thinking about it, this site here, Juneau, let's keep that site in mind for a minute. This is one of my favorite sites. I think it's a really beautiful spot. It has really diverse algae on the site. It's got some hard rocks surrounded by boulder and cobble. Really cool site, but really low survival. And at one point, we even decided to take this site out of the network, and we stopped overseeding to it, which was really unfortunate. My daughter's nickname is also Juno, so that may be why it's one of my favorites.
Is the density that you're seeding the sites with, like, how many abalone is there?
Yeah, in a given year, so we're not seeding every site every year. We're kind of in a 3 to 4 year rotation. So the good sites will get overseeded every 3 to 4 years. And each time we outplant, we add anywhere from 500 to 1,000 animals per site.
And how big is the site?
They're roughly 8 by 10 meters. So the density at outplant time is pretty high, but those are all very small animals that, many of which won't survive. So I just wanted to hit a few takeaways from that paper. The first is that our early survey results are actually really good indicators of whether or not a site is going to be a success or a failure. Early on, we would see low survey results in year 1, and we would like a site and we would just keep overseeding it. I think we're realizing that that was probably a mistake. If we see in year 1 that survival is low, we need to assume that that site is probably not going to work out for us. So we're no longer outplanting to sites that have less than a 2% success rate. And that's specific to that metric I talked about, looking at reproductive size over total of number of abalone outplanted. Second thing I wanted to mention is that we need to quickly replace the failed sites with new sites, and we need to overseed the good sites to continue building density and diversity. I already mentioned that our detection during monitoring is low. We think it's 20 to 30%. And this is a big one. What affects juvenile survival the most? It's not the size at which the animal was outplanted. It's not the genetic family that it came from. It's actually site. Site is our biggest driver right now in why a site succeeds or fails. So this is kind of our primary constraint to the recovery efforts. So we need to identify sites with potential for high survival. So how are we going to do this? This is a big one, and we're really right now we're trying to tackle this question in 2 ways. The first is these oceanographic sensor arrays. We're starting to wonder if maybe there's really small differences in water chemistry or oceanographic conditions from one site to another that might be driving survival. We've seen sites that are not even a mile apart, and one will do really, really well and one will fail. So what's driving that? To get at this, this work is being spearheaded by Eileen Bates. She's at the University of Washington SAFS program, a PhD candidate. She is doing a really tremendous work, not just on these oceanographic sensors, but on other areas of our recovery program as well. So she's actually developed these, I'm just gonna go back here. So this is a picture of the sensor array. So this right here is a flow meter, and then this includes pH, dissolved oxygen, temperature, salinity, light penetration. I think that's all of it. So all those parameters are being monitored continuously. She's also taking microbiome samples. So at each site, we're collecting kelp and algae, and then looking at the kind of the microbial community at that site and doing analysis to see if maybe are differences there from one site to another that could be driving survival. We've got these arrays on 6 different sites. We're 2 years into the project, so we'll start our 3rd year on 6 new sites in February. Every 2 to 3 months, we have to be up there recovering those arrays, downloading the data, calibrating, and then reinstalling those sensors. So it's a tremendous workload. And ultimately, we hope to answer, whether or not there's a correlation between sites that are doing well and some of these oceanographic conditions. Fortunately for Eileen's research, unfortunately for the recovery program, the first year that we did this, we had a really good spread of high survival and low survival across the sites where Eileen's sensors were installed. Same thing in year 2. So that could really be helpful in determining, are there differences that we can correlate to that success and failure. Very cool study. I'm really excited that that's underway.
Are you being more broad? Like, sounds like a lot of factors, but she really spread it out.
Eileen's work is all on our recovery sites in, actually, yeah, San Juan, Skagit, and Island County. So her arrays cover sites across our network, not just the San Juan Islands. No Jefferson.
I mean, I just know other science people I know around here say like Spin Bay is in really good shape.
Yeah, there's so much ground to cover and so much water, so much shoreline. And eventually we'll be moving west and doing more of this research in more places, but we are limited to San Juan, Skagit, and Island counties. Our sites in Island County are at Smith Island, which is a really cool, dynamic place. It's the biggest kelp bed in Washington State, and there's some really neat stuff going on there. I mean, kind of getting off on a tangent here, but there's really a lot to learn yet, even about abalone specifically in the Strait of Juan de Fuca. We haven't been able to do the same type of really thorough, index station surveys in the Strait. A lot of what we know about abalone in the Strait of Juan de Fuca is based on these 10, 20, 30 years of urchin index data. And those same surveys are, abalone are counted in those surveys as well. And that's kind of what we know about abalone in the Strait. So yeah, I'll kind of get back on track here, but yeah, there's a lot of work to be done in other places here in Washington. So the other thing that might be driving success and failure at a site is this idea of predation. Are there certain sites that just have a higher, load of predators? And we're getting at that with these time-lapse cameras. So we're using a sub-aqua imaging systems. It's built by a company in San Diego. It's got an underwater camera and strobe. We have 6 cameras total. So at any given outplant time, we'll monitor 3 sites, and we put 1 camera on plot, and we put the second camera off plot by 5 to 10 meters as a control. And that camera is also set up on empty outplant modules with no abalone. So trying to get a sense for if the critters that are there, are they there because of the abalone, or are they just there? We take an image every 60 seconds, and then the cameras are removed 48 hours post outplant. To give you guys kind of where we stand so far. So we've done this 3 years now, and we're going to continue doing it. We have over 100,000 images from these cameras. And we're doing an analysis of these images, looking for what's there: fish, sea stars, crab, octopus, other predators that we may not have known about. And so far, there is no incriminating evidence. And by that, I mean, we have yet to find an image where a crab has a juvenile abalone in its claw, or a fish has an abalone in its mouth. It also seems that there's really no statistical significance to a certain number of animals being at one site versus another that has survived better than another. So really, there's nothing happening in terms of predation that we're seeing from these images. There is an abundance of kelp greenling, which we obviously see an abundance of kelp greenling, but they seem to really be drawn to our outplant modules. So initially we thought for sure these greenling were having an effect by preying on these small abalone crawling out of the tubes. But we've also seen really high loads of greenling at sites that have survived really well. So again, no statistical significance there. There doesn't seem to be any connection between a lot of greenling and loss of abalone at a particular site. In terms of those controls, again, those greenling are swarming the control modules just like they are the treatment modules on the outplant sites. So it seems like it's more a curiosity rather than hunting. And then the last takeaway is that it's become clear that the juvenile abalone will quickly leave these modules as soon as we remove the screens, and they're crawling down into cracks and crevices and finding that protective habitat. Yeah, I want to wrap things up here so we have some time for discussion, but these are just a few of the images. I know it's a little hard to see, but I tried to capture some of the ones that I thought were more interesting. This is a helmet crab, not really a predator of juvenile abalone, but we have seen these guys crawling through the image a lot. This is a sharp-nosed crab, and this guy seems like he might be interested in what's going on inside this outplant tube. But again, no incriminating evidence. Here's an octopus. This is the only octopus that we have seen in over 100,000 images, and that was a big surprise to us. We thought that octopus could be a prime suspect, and that's not the case. And then this column here is all kelp greenling. They're hard to see, but you can see this guy is kind of checking out the tubes. He's got his mouth wide open. This guy's underneath the tube. Also seems like he's really curious about these little abalone that are crawling out. And then again, kelp greenling kind of right with his nose right up against the tube. I don't want to run out of time, but one of the more fascinating things that we've looked at recently is the genetic implications of what our program may be having. I'm not a population geneticist and I'm not a molecular biologist. So this is a great paper. And if you are a population geneticist, I would say take a read at this paper. It's really fascinating. We worked with Dr. Jay Diamond at Western. He did a postdoc at PSRF, and he was a tremendous resource to actually helping us answer these questions that have been in my mind for 10 years. You know, what's the diversity of that broodstock population that we're taking from the wild? And, you know, what's happening with families that we outplant to these recovery sites? You know, clearly not all of the families are surviving. If we put 50 families on a site, are there 12 that are surviving to reproductive size? And that may not be a good thing. And what's the relatedness of the animals that we're producing? And is there potential that these family groups that we're producing could be, you know, spawning on these recovery sites? So all these questions about the genetic implications of our program have kind of been swirling for a decade. And Jay really tackled this one and allowed us to answer these questions. So we actually took 457 tissue samples from 8 different populations that included kind of our historic broodstock population. So these are animals that were collected and brought to the hatchery over a 10-year period. We looked at a smaller, newer group of animals from the San Juans. We actually had a batch from Ketchikan, so something a little bit outside our range. And then we sampled from our 4 most productive outplant sites, and this was really difficult to do. You know, are we going to have a negative impact on these sites that are doing really well by going out there and taking these tissue clips? I was nervous about it, but the risk was worth it. We have a non-lethal sampling method, and we were actually able to get, I think, 250 samples from these 4 sites, maybe more. And then we also included a population from our most 4 or 5 recent hatchery cohorts. So we use genotyping by restriction site associated DNA sequencing. It's called RAD-seq. And Jay was able to identify 49,000 SNPs and 8,000 haplotypes. So those are basically just markers. And you can use those markers to make a comparison between individuals and populations. In the past, microsatellite markers have kind of been the way to go for population genetics, but for abalone, there's really only 6 or 7 markers to use. If you choose microsatellite markers, you compare that to, you know, over 50,000 markers that are going to help you understand those differences between populations. So RAD-seq seemed like a really good way to go for this study. There's a lot of figures in this paper. I just picked this one because it kind of describes the allelic richness. And that's really just a measure of population diversity and is indicative of how well a population might be able to adapt and persist over time. So the higher this number is, you know, the better chances that that population has. This came out exactly as you would expect. These top 3 lines, that's our wild, those are basically our 3 wild collected groups. These bottom 5 are the 4 outplant sites, and then that 1 new hatchery cohort. So as you would expect, there's more diversity in wild populations than there are in what we're producing for the outplant sites. But the study was still a really good indication that what we've produced so far is a really good representation of wild populations. Even though that allelic richness was a little bit lower, overall, what we've put out there is a really good representation of wild populations. I'll just kind of skip through this since we're out of time. But the other thing that was neat about the study is that Jay found that there's very little chance that related families that we've put out there are spawning with each other. So that's a good indication. And then lastly, we're going to use that same technique to do an analysis of range-wide population genetics from Sitka to Baja, which will be really neat, depending on what steps we need to take moving forward in this recovery program. Hopefully, we don't get to the point where we need to import broodstock from out of state. But if we do, that range-wide study will be a really key thing. All right, it's almost 4 o'clock. There's a lot of other research happening related to ocean acidification. We've worked on sperm cryopreservation in the lab to try to give us one more advantage in the amount of diversity that we can produce. We have kind of shifted gears from outplanting 2-year-olds to 1-year-olds to try to get animals out of the hatchery sooner. That seems to be going really well. We've looked at larval seeding as another technique, as opposed to outplanting juveniles that are about 1 year old. So there's a lot happening, and I wish I had more time to dive into all of it. Basic next steps. We need to secure long-term funding so that we can fully implement the recovery plan that we've developed. We want to continue building these network of recovery sites, like we said, not just where we are now, but continuing to move across their range. That's going to take a ton of partners, and that's why it's exciting to have the Seattle Aquarium team on board this year to start tasking these recovery efforts to other organizations and dive teams that are capable. We want to continue to expand our captive rearing capabilities. Having those satellite nurseries is key. Thinking about ways to expand those, as Port Townsend Marine Science Center develops new facilities and systems, just always thinking about ways to expand that capability. And lastly, there's a ton of research happening, and we need to apply that research to our conservation efforts. That was a lot to get through. Thanks for being patient. There's a ton of people to recognize, or a ton of funders to recognize. So I just want to add that. And lastly, I would love to take questions.